Hi, I'm Peter, and this is my presentation on reporter assays. So here are some learning objectives. This is what I kind of want you guys to get from this presentation. First of all, what are reporter assays and how do they work? Second of all, let's learn a little bit about the types of reporter assays. What are the pros and cons of each type of reporter assay? And thirdly, how are reporter assays used and applied in research? So we're all biochemistry majors or biology majors or some related field, and I'm sure we all know how important and how often the word assay comes up and how important assays are to our field. But if you're like me, you haven't actually done one or really delved into the complexities of one since biochem lab. So an assay is an analytical procedure used to quantify or qualify the amount and or activity of a target of interest. More specifically, enzymatic assays are one of the most common tools in molecular biology. They have big applications in drug discovery, disease diagnosis, genetic design, screening, and more. Most enzymatic assays are quantified via detection of fluorescence, bioluminescence, or photometric analysis, such as absorbance spectroscopy. This image down here is a burette assay that we did in Biochem Principles Lab last year. You can see we ended up quantifying this one through absorbance spectroscopy. Now let's get into a more specific type of assay, the reporter assay. A reporter assay is a type of assay that is commonly used to study the transcriptional activity and or regulation of a gene of interest. It also has other uses, but we'll get into those later. The mechanism of a reporter assay is really through the reporter gene. A reporter gene codes for a protein with a unique quality that allows for easy detection and quantification of that protein. Associating a reporter gene with a regulatory sequence of the gene of interest enables for visible detection of regulation activity by a promoter or some other regulatory mechanism. There are various types of reporter genes that allow for various applications of reporter assays. This image down here shows a simplified view of a reporter assay. As you can see, a reporter gene is associated with a regulatory sequence of interest. So this is a regulatory sequence associated with a different gene, but that gene is cut out and replaced with the reporter gene. And this reporter gene is transcribed into mRNA and then translated into a fluorescent protein that is easily quantified. Let's look at a little more in-depth view of the mechanism of reporter assays. So generally, the reporter gene is incorporated into the genetic sequence of interest downstream of the endogenous regulatory sequence. Now, depending on the demands of your study, the endogenous gene, the gene whose regulation you are studying, may be cut out completely. This is because that the protein, the endogenous protein that this gene codes for, may interact with the reporter protein that your reporter gene codes for, and these interactions may cause deviations in your data. This is shown here in this figure as a target regulator is cut out from its endogenous coding sequence, and it is attached to the coding sequence of a reporter gene. So the reporter gene is now fused with the endogenous regulatory sequence. As a result, it will be expressed just like the endogenous coding sequence would normally be expressed. As this is summarized below, target cells are then transfected with the hybrid genes, and the cell environment can then be manipulated based on whatever study you're doing, and the measurable reporter gene expression will be easily detected just like it was the endogenous gene. As I mentioned before, there are a wide range of reporter genes that code for a wide range of reporter proteins, and this allows for many applications of reporter assays. So the first specific kind of a reporter assay that we're going to get into is enzymatic. So an enzymatic reporter assay utilizes a reporter gene that codes for an enzyme that can produce a measurable change in a substrate. For instance, changing a colorless substrate to a colored substrate. The most common enzymatic reporter gene is bacterial LAC-Z, which codes for beta-galactosidase. Beta-galactosidase catalyzes many reactions and it acts on many substrates, but the main one that's used in reporter assays is the reaction of O and PG. Beta-galactosidase reduces ONPG to its ONP anion, which has a bright yellow color, which can be measured via absorbance spectroscopy or other spectroscopy methods. And this measurement can be related directly back to the level of protein expression. Like I said, LAGZ is a very versatile gene, as beta-galactosidase can act on many substrates. However, it is not optimal for mammal cells because beta-galactosidase is endogenous. So using it in mammal cells, you would be required to remove all the endogenous beta-galactosidase so that it doesn't interfere with your assay. And shown on the right is just a visualization of what happens when that regulatory sequence is on and it's coding for your reporter protein that changes the color of your substance, and this can be measured. So another very common form of reporter assay is a fluorescent reporter assay. We'll focus on green fluorescent protein specifically, also known as GFP, which is found in a species of jellyfish. When GFP is excited with 395 nanometer light, which I think is violet, it emits 509 nanometer light, which is a very bright green. And the intensity of this light can be measured and correlated directly with the expression of the protein. A pro of GFP and using fluorescent reporter assays is that it doesn't require an exogenous substrate. All you got to do is shine it with this specific wavelength of light. However, these assays are not as sensitive and not as accurate. And here, this image is the classic form of GFP. It's got that barrel structure that is directly related to how it is able to emit light. Another type of reporter assay utilizes bioluminescence. It's technically an enzymatic reporter assay, but the enzyme that the reporter gene codes for doesn't produce a measurable change in a substrate. It catalyzes a reaction that releases light as a product, and this is bioluminescence. This light that is released is then measured and correlated to the expression of the protein. Firefly luciferase is the most commonly used reporter protein in these types of assays. This is obviously the protein that makes fireflies glow. Firefly luciferase oxidizes the compound luciferin, which produces a quantifiable flash of light. Now, a pro of this assay is that it's highly sensitive and highly accurate, but a con is that its flash is very short, and there are actually reagents that you may have to use if you're using this kind of assay that lengthen the half-life of this flash so that you can measure it. So the final type of reporter assay that I'm going to cover are epitope-based. So what is an epitope? Also known as an epitope tag. So an epitope tag is a short polypeptide sequence added to either the N or the C terminus of a protein. And by doing this, it tags the protein. These epitope tags are recognized by antibodies, which allow for the detection of a protein that would otherwise, like all these other ones, be difficult to analyze. These antibody epitope relationships can be detected via immunoblotting, immunofluorescence, or immunoprecipitation. A pro, much like the ELISA assay, which uses antibodies, is that this is a highly, highly specific assay that will provide accurate and sensitive results. This figure down here shows the process of incorporating an epitope tag into your protein. As you can see, the short polypeptide sequence, or the coding region for that polypeptide sequence, is incorporated after the gene of interest, and the resulting protein is tagged with this short polypeptide sequence. And then this shows two different methods of quantification: blotting and immunoprecipitation. So as I mentioned before, there are many different applications for reporter assays. Listed here are just some of them. For instance, you can use a reporter assay to determine where a specific gene is expressed, such as in which type of cells. This is often utilizes GFP, which can actually tag a protein and make the protein fluoresce, which we will talk about a little bit later. You can use reporter assays to interpret the effects of a condition, drug, or small molecule on gene expression. This is where you would cut out the endogenous gene completely and just fuse the endogenous regulator with the reporter gene. You could use the same technique to evaluate the strength of a promoter of interest. And then another way that reporter genes are utilized are under control of their own regulator, of their own promoter. They are transfected along with a protein of interest or a vector of interest that you actually want to transfect cells in. But as they are transfected with, say, for instance, GFP, whatever cells fluoresce, were successfully transfected with the vector that you're interested in. And then also, what we're going to get into a little bit later, it can be used to track protein within a cell. So here is a real application of reporter assays in research. This is a paper I found produced by some researchers in Poland, and they produce a luciferase-based reporter assay to determine the effect of certain small molecules that they isolated from cyanobacteria on cancer-linked microRNA. So a quick background on microRNA. They are not coding RNA, but they are often regulatory molecules. And this specific microRNA that they were focused on was defects in it were directly linked to liver cancer. So here in the image, you can see their setup. They included a luciferase gene downstream of a regulator that was under the control of this certain microRNA. They believed, or they predicted, that if the small molecule was successful in binding the immature, the microRNA, that the RNA would not be capable of inhibiting the regulatory sequence that is upstream of the luciferase. So what would the result be? It'd be a very bright luciferase signal. Similarly, if it wasn't successful, the microRNA would be allowed to mature and therefore inhibit that regulatory sequence, and there would be a dim, if any, luciferase signal at all. While they did not determine that these were cancer-curing small molecules, they did present this reporter assay as a reproducible system for this kind of research. So here's another example of how reporter assays are used in research. This is on the parasite Leishmania. I hope I'm saying that correctly. And Leishmania is a parasite that causes the disease called leishmaniasis. And the interest of this research is that they know that Leishmania does not produce its own heme. It must steal it from its human host. So a study by researchers at Oregon Health and Science University identified and characterized this protein, FLCVRB, which is the first known heme transporter of Leishmania. So they assumed that after the heme was incorporated into the Leishmania cell via endocytosis, this is the protein that would carry it around. But they had to prove it. And they used GFP tagging to pinpoint the location of this protein within the parasite under different endocytotic conditions. So here is an image from their research that I think really summarizes what they were able to do with their GFP tagged protein. So here the reporter assay was just used to localize where the protein of interest was at in the cell. It's under two different temperature conditions. On top is 4 degrees Celsius, which is not prime endocytosis conditions for Leishmania. So no endocytosis was going on, but you can see that the protein that they assumed was a transporter for heme was located at the cell membrane and the flagellar pocket. However, once endocytosis conditions were reached at 28 degrees Celsius, you can see that the protein was also found in an intercellular pocket, which is a known part of the endocytosis route of this parasite. So that pretty much proved that this molecule was picking something up during endocytosis and taking it into the cell. So here's just a quick summary of reporter assays. The general idea: incorporate a gene that codes for a protein with a unique measurable quantity with whatever you want to study, whether it be a regulatory sequence or another protein. And the resulting protein that can be measured as if it was just the endogenous protein. Detection is sensitive and simple, and it's just achieved by measuring fluorescence or bioluminescence with absorbance spectroscopy. And finally, as I've been emphasizing reporter assays are extremely versatile. With many different types of genes come many different types of applications. Thank you very much.